Thanks, guys. Good afternoon, everybody, and thanks for the organisers for the opportunity for us to present some of this work, which, as has been said, is a collaborative work between ourselves at CM Bio and the group of applied regulatory science at the FDA. So I think everyone knows that as doing preclinical biology now, there's a really wide range of tools available, and most prominently, the rise of MPS has become a really key part of this toolbox to complement a wide range of other tools that are available. And picking the right tool out of this toolbox is key if you're ultimately going to get the translation that you need to predict what's going to happen in a human. And so how do we model the human liver? Well, the human liver is a key metabolic organ. It's the site of most common forms of drug toxicity. It's the key site where drugs are metabolized. And there are obviously a bunch of ways to model a human liver. Well, you can go and get some mice or some dogs. This is often used for modeling liver disease. And for many, many years, we've cultured primary human hepatocytes in plastic dishes. And typically, primary hepatocytes don't like that. And their functionality disappears after just a couple of days, trying to culture them on 2D plastic, even on collagen. So people have now been growing these in 3D and spheroids, sometimes as co-cultures, and this improves their functionality and is very, very nice. But can we go one step further? And this is where MPS organs on chips really comes in. And another novel technology that's also recently been developed is the use of precision cut liver slices. So how are you going to make a really good MPS to model the human liver? Well, the industry IQ MPS consortium a couple of years ago did a really great job of articulating this for the community. And I'm sure a number of you are aware, a group of lab on a chip papers was published describing some of the key criteria for what a liver MPS should be able to do. And so these include the production of the key biomarkers, the expression of key enzymes and transporters, and the right signals that a liver MPS would produce in response to damage. And so this is a real framework now for developers to work off to describe how we should build a successful liver MPS system. And there's been a whole bunch of liver MPS systems been built over the years. And probably another we're going to get described at this conference. And one of the key challenges still remains, as was talked about in some of the previous conversations, was, well, how do we drive the adoption of these technologies? How do we make them being used every day? And one of the key activities is to engage with the regulators to make and to do cross-lab into an intra-lab comparisons to show that these are reproducible, robust tools that can be used every day to ask key questions and to do this in a context-dependent manner. So let me show you more about our liver MPS that we have at CM Bio. So this was originally developed in Linda Griffith's lab at MIT a number of years ago, and we've taken this on and developed it further. So we have quite a unique MPS system. It's an open well-based system, looks very much like a cell culture plate. It has 12 individual wells. Each one of these wells is individually perfused, and the media recirculates through a three-dimensional scaffold that's held within the fluid path. And what we do is we seed single cells into this scaffold. These are typically cryopreserved primary human hepatocytes. They go in, and over a couple of days, these single cells form three-dimensional microtissues within the scaffold. And actually what we have is about 300 of these microtissues arrayed across a scaffold. So we basically recapitulate sort of mini columns of to mimic the liver microarchitecture. And so what these kind of look like, and these can be hepatocytes, or I'll show you in a minute how we can incorporate other cell types of the liver within these three-dimensional perfused microtissues. And that perfusion of media continually through the scaffold, through these microtissues, is really key to give the cells their functionality and maintain their functionality, which they lose in typical 2D culture. So we get these beautiful tissues. They produce key biomarkers like albumin. They have bicanaliculi. They have tight junctions, and all the fluidics is controlled by our control system that you see there on the screen. And so a typical experiment with this setup normally takes a couple of days to seed those cells in, and we then have a really pretty long, oh, wait a minute, why do I keep going forward? We then have a pretty long window to run experiments in. So this allows us a lot of opportunity to do a wide variety of different studies, either in the tox or efficacy space, to really evaluate how liver biology is changing following an insult. And this model typically uses, as I say, primary human hepatocytes, but it's not limited to that. We use the system with stem cell-derived materials or even clinical trial samples now to recapitulate their function. And we can do a wide variety of endpoints, as I'm going to show you to assess liver functionality. So is this model actually functional, and does it look like a real human liver in some way? Well, the answer is yes, it does. So if we take the bulk RNA-seq data from these chips, and we can do this longitudinally over time, and we ask like an online portal what it thinks it's looking at, it's probably pretty difficult for you to see slightly, but it thinks it's looking at bulk liver tissue come straight from a patient. It thinks it's primary hepatocytes, and all the key markers and key genes, the CYP enzymes, etc., are all still expressed, albumin being one of the most abundantly expressed genes. We express the key biomarkers of the liver, so those same biomarkers that were described in our IQ MPS paper, we produce in the system, we produce them to exactly the levels that were described in that paper, and were pretty much what your human liver would produce in any one of you guys. And importantly, we have metabolic activity. So from a drug metabolism perspective, we've got to express all the CYP enzymes that are present, and we can do this across multiple donors of cells, pools of donors, etc. And as I alluded to earlier, these microtissues primarily need to be formed of hepatocytes, but they can incorporate a whole range of other cell types too. So we can typically incorporate Kupffer cells, and I'll come back to this later. To have an immune component, we could add stellate cells, and we can see all the markers of those being expressed if we need to. We could also even add peripheral immune cells into these chips and allow those immune cells to perfuse around if we want to. So we've taken this liver MPS and put it in a whole bunch of different applications over the years. We have a wide variety of publications on this, ranging from work on DMPK and ADME, which I'll touch on in a minute. We've done a lot of work at disease modeling. I've got a lot of expertise in building models of human liver diseases, particularly of fatty liver disease, NAFLD, NASH, and of viral hepatitis. And we've used the system extensively for looking at drug-induced liver injury, which is still a really big key issue and a clear context of use where liver MPS can add a lot of value. So let me tell you a little bit more about the collaborative study that we did with the group at CEDAR in the FDA. So this came off the back of a DARPA-funded program we did along with Linda Griffith's lab, and we got interacting with the FDA, and they were very keen to see the outputs of that DARPA-funded program and the technologies that have been developed, and could they really be evaluated in much greater levels of detail and shown that they are reproducible and ready to produce IND-like package data. So what we did was we got the system set up in the labs at the FDA, and we have the system obviously set up in our own labs, and we could then cross-validate between those two labs. So first of all, we started pretty simply. Well, could they form the same consistent liver microtissues? And the answer is, well, yes, we could. See the standard sort of images there. Could these cells are also metabolically active? So this is the CYP activity. And these microtissues, we specifically decided to have an immune component, so they all contain primary human Kupffer cells. And so when we add an immune challenge, an LPS challenge into these microtissues, that metabolic activity goes down, just like it does in the human liver. And very importantly, I know these systems could reproducibly produce these microtissues. This is a really key attribute that the FDA are interested in. And if these technologies are going to be widely developed, any end user really wants a reproducible, consistent system that they can use over and over again, and again with multiple different sources of primary cells from different donors. And we assess the reproducibility. We assess the reproducibility of the system across different cell sources. What we then did was then take the same cells, the same combination of hepatocytes and primary Kupffer cells, and put them into the liver MPS, and at the same time put them into spheroidal culture and 2D matrigel or sandwich culture, and asked, well, are the same cells like functional in all of these systems, and can they maintain their function? And what we could clearly see was that in the liver MPS, the same cells were much more metabolically active. They produced much greater levels of albumin, and this was maintained over a long period of time. The standard spheroidal and 2D sandwich cultures, I know at the beginning, for the first few days of the culture were okay, and they were doing as you would expect. But over, 3, 4, 5 days, that functionality and that metabolism really dropped away. And you do see a slight decrease. Oh, I keep pressing the wrong button. You do see a slight decrease in the metabolism over time, but this is still way above what could be achieved in any of the other systems. And exactly the same was seen for albumin. So we really see that the liver MPS is creating the right microenvironment to maintain the phenotype and function of these cells. So then we really wanted to understand, well, can this be used for key context of use that the pharma and drug development industry cares about? And so we chose two different areas to look at. The first one being DMPK and pharmacology, and the second one being safety, pharmacology, and toxicity. So to assess DMPK, could the system be used to assess the clearance of human drugs? We looked at the pro-molecule diclofenac, which has been well used and well studied for this type of application. And what we could see, if we dosed diclofenac into the system without any cells, it was very stable in the system, it didn't go anywhere, but as soon as the system contained those liver microtissues, it was rapidly turned over and cleared away by the liver microtissues. And this is exactly what you'd expect when this drug is dosed in vivo. Now, another small but very, very important piece of data that was generated was to show that the system has really low, almost minimal non-specific binding to these small molecules. This is a real chronic issue that's plagued the MPS industry for a while. The use of soft polymers are well known to absorb small molecules in particular, and also can absorb a whole range of different proteins too. So we did a nice study, part of this, we took a whole bunch of range of pro compounds, we put them in the system, we showed that they didn't bind and don't get absorbed. So this enables these DMPK studies to become much more straightforward. The other key thing that the DMPK study wants to be able to do is to see the production of human-specific metabolites and understand the breakdown products of any particular compound. So here we use the example of triglyceridone. We could put this into the system, incubate it with the liver microtissues, and we could pick out the primary and secondary metabolites for triglyceridone being produced. And this is key because you've got human-specific enzymes here, so you'll get the human-specific readouts that are coming out of this system. So we could demonstrate that the system was very applicable for DMPK type studies. Well, could it be used for toxicological assays and understanding how could you predict the toxicological response of a compound in the system in the same way that you do or you more standardly do in 2D or spheroidal cultures? And again, the answer was yes. So we used a range of pro compounds. They were all, again, run in parallel through the three different models, the liver MPS, the spheroid, and the sandwich culture. And we could look at a wide variety of endpoints of biomarkers. We could see decrease in CYP activity and albumin, dose-responsive effects. We could see the production of and release of LDH. And all the different systems were able to produce IC50 curves, which is a key fundamental of any liver toxicological assay. But we wanted to go a little bit deeper than that, and could we show a real elegance and a real depth to what the liver MPS could do? And so we used the example of trovafloxacin and its twin molecule levofloxacin. And trovafloxacin was removed from the market for causing idiosyncratic DILI. In the presence of inflammation, it was found that trovafloxacin, the antibiotic, would cause real severe liver damage in some patients. And so we asked, well, could we recapitulate this in the system? And to walk you through this data, the answer is yes, we could. So we dosed both levofloxacin and trovafloxacin in the presence and absence of an inflammatory stimulus, so here LPS. And we see that the trovafloxacin causes significant DILI, so we see the release of LDH and the reduction of metabolic activity of these cells. And this was driven and enhanced in the presence of the lipopolysaccharides. So in the presence of the inflamed, damaged liver, we get much more susceptibility to DILI, and this is exactly what is seen clinically. And importantly, to really add to the reproducibility piece, this study was done with multiple lots of cells, which is key, and it was also done at multiple sites. So we could do exactly the same study, the same system, and really demonstrate the kind of reproducibility and robustness that you could get this data wherever you are in the world and in whatever lab. So in summary, hopefully I've shown you this afternoon that we've built a liver MPS system that is really robust and ready for use for a wide variety of different uses. The liver microtissues that we form are really, really highly functional and can be used to assess a whole variety of different clinically relevant biomarkers. The microtissues can be very consistently formed from different sources of cells to give you really high content data. It was just mentioned by one of the previous speakers. These systems are really high content. These not really HTS screens. They need to complement or work alongside HTS screens to get the right set of preclinical data that you want to ultimately be successful. So thanks for listening. We have more information about this MPS system across the week here at the MPS World Congress. So we have a number of posters and we have a booth. You can come and see the system and play with it yourself. So thanks for listening, and I'll take any questions.
I'm Donna Mender from NCTR FDA. So I have a question. At the beginning, you mentioned you can use primary hepatocytes, cell lines, iPSC. Have you done that kind of comparison where you looked at metabolic activity over time and other aspects to see which incoming cell type is most important?
Sorry, I think I only caught half that.
Okay,
was your question, have we done HEPs, IPS, and cell lines in parallel?
And looked at metabolic activity over time and the other analysis you did with primary hepatocytes. I'm just wondering how the other cell types acted.
Yeah, so the system is totally compatible with all of those other cell types. We see enhanced functionality of those other cell types in the MPS compared to static 2D cultures. Those cell types don't have the same metabolic capacity, particularly of the primary hepatocytes. They just simply don't have the horsepower. IPS particularly, I think, are the future to some extent, and I say that with a sort of wince, because of their unlimited source, their kind of ability to genetically control them. I think we really want to be able to build all sorts of MPS using iPS-derived cells, but their functionality is still not the same level as primary human cells. So the focus for building MPS at this point has got to be on those primary cells. And whilst we work with the IPS community closely, because IPS cells or IPS-derived cells in these type of systems do function much better, but they don't suddenly become fully mature just because you put it in MPS.
Okay, thank you.
Any questions? Well, thank you for your talk. I have a small question, if I may. So how did you quantify reproducibility in your MPS?
That's a great question. We really just looked at the ability for the microtissues to form across multiple wells, multiple plates, as I say, across multiple sites, and with multiple cell sources. We didn't do a deep statistical analysis to determine how specific numbers that we should look at. But when we looked at across a range of plates, range of site range owners, we see greater than 90% of all the microtissues are the same. And I think that, but what's important, that is done with cells that we know will work in these three-dimensional tissues. There's lots of primary cells out there that won't form good quality three-dimensional tissues in any MPS or even any spheroids. So I think I should qualify that these cells were pre-qualified to do that. And once you do it, you then get that high level of reproducibility.